Greetings, respective judges and fellow peers. Today we are going to present our review entitled Employing Omics in Cancer Drug Discovery, Recent Updates Towards Personalized Medicine for Lung Cancer. But before that, let us introduce ourselves first. My name is Ciputra and two of my colleagues are Nathaniel, Alvin, Sanjaya and Alvaro, Filbert, Liko. Lung cancer is still the leading cause of cancer-related death worldwide. It poses high risk of complication followed by ineffective treatment in some of the patients. The key problem in lung cancer management lies in the fact that lung cancer has tremendous biological heterogeneity among populations or even individuals. Histologically, it is simply classified into two main subtypes, small cell lung cancer and non-small cell lung cancer. However, molecularly, it has various genetic alterations. It is why general therapies are not effective in all patients, since different genotypes will lead to different phenotypes, thus demanding different treatment modalities. The development of specific treatment for lung cancer is also supported by the advancement in molecular technique, known as omics-based analysis. Omics-based analysis integrates biological science and data science to provide a comprehensive molecular and clinical analysis of biospecimens and biological data. It consists of multiple layers from genetic, transcriptional, and translational level to even higher level omics such as cytomics and interactomics. The ability of omics technology allows optimization in reverse pharmacology research method, which relies on specific molecular signature to develop patient-specific treatment. Omics technology can be used to identify the patient-specific target by mapping the molecular signature of the disease, providing sufficient information for drug development, which will be to bedside after being validated. By doing so, we look forward to a precision medicine strategy to develop a more appropriate and selective treatment for lung cancer. But before that, we need to develop a standardized lab for omics implementation in reverse pharmacology research, which is currently lacking. Therefore, in this study, we aim to identify and review the molecular signature of lung cancer based on omics analysis and currently available targeted therapy to construct the standardized pipeline for omics implementation in lung cancer targeted therapy. To do that, we perform scientific literature review by using Google Scholar Search Engine. The keywords used are lung cancer, omics, and targeted therapy. Research paper, literature review, short comments, systematic review, and meta-analysis published in the past 10 years, which are relevant with current targeted therapy for lung cancer and molecular signature of lung cancer based on multi-omics analysis will be selected. Then a general pipeline for omics implementation in lung cancer targeted therapy was constructed and discussed for its optimization in Indonesia.
For the result section, the omic-based molecular signature found are classified according to the histological classification of lung cancer. The first one is the non-small cell lung cancer, or NSCLC in short. The most observed and reported molecular aberration in NSCLC is the one involving the epidermal growth factor receptor, or EGFR. This aberration, for example, could lead to uncontrolled cell proliferation due to continuous activation of the receptor that indicates cell proliferation signal. However, as we can see here, the lung cancer cellular machinery might not be as simple as that. Through multiple omics study, we can see the bigger picture, that different lung cancer patients might host different subtypes of lung cancer that have diverse molecular signatures. Small cell lung cancer possesses an aggressive histological subtype. Effort in mapping its molecular characteristics are still lacking due to the low prevalence, aside from the fact that it is the most common type of lung cancer in smokers. The consequence of this molecular aberration can also be linked to hallmark of cancer, similar to the one in NSCLC. Based on the omics molecular signature, we can conclude that the lung cancer has complex molecular aberration. The next question is, how can we address this? Due to the time constraint, we have summarized the table from our result section regarding the currently developed targeted therapy for lung cancer. The key points are the following: that the focus of our current targeted therapy are still in EGFR, which lead to subsequent problems such as resistance. Another point will be that all of the drugs developed only use single omics perspective. thus allowing eventual escape of the lung cancer itself. Finally, an interesting finding from our result section is that all of the natural-based drug research has never reached even clinical trial, indicating the lack of optimization in ethnopharmacological research output. So here are the table that we have summarized the points from. As we can see here, in this slide, some of the natural-based drug research product that we have actually found. As we can see, they have actually multiple targets in lung cancer therapy. However, as I have mentioned earlier, none of them actually have reached the clinical trial step, which indicate lacks of safety and efficacy.
Moving forward to the discussion part, we have seen that single-level omics-based analysis is not adequate to achieve an effective treatment in lung cancer patients. Therefore, multi-omic studies integration is important to develop better targeted therapies as seen in our developed pipeline. For instance, we can collect several biospecimens from patients, such as tissue biopsy, plasma, or urine. Using multi-omic analysis methods, we can gather various biological data, such as mutations, gene silencing, gene expression, protein trafficking, metabolic pathways, and cellular markers, which in combination may help in determining the most appropriate treatment for the patient. Omics-based analysis can also be applied for other biological data that are not directly obtained from biospecimens, such as histological and radiological images, which are commonly used in lung cancer diagnosis. As shown in this figure, bioinformatics analysis, such as stratification and deep learning systems, can be conducted on biological data, and this can be translated into data-driven predictions for more precise patient management. Omics-based analysis may also allow continuous monitoring and treatment adaptation of patients. Since tumor profiles are susceptible to changes after treatment, it is important to evaluate treatment response by reassessing the molecular profiles. By doing so, administration of ineffective treatment may be minimized, thus reducing unwanted costs and improving patients' quality of life while preventing tumor escape. Last but not least, omics analysis may also be applied in Indonesia's ethnopharmacological research. By integrating our natural product library with Indonesian population-specific molecular profiles, we can employ natural products as novel, alternative, and promising treatments for lung cancer. To summarize, omics-based analysis has huge potential as our primary vehicle in alternate targeted therapy development for lung cancer by shifting drug targets from just tyrosine kinases to the complex molecular profiles. Integration of natural product libraries with Indonesian population-specific molecular databases is the main key to optimize Indonesia's biodiversity for better ethnopharmacological research for cancer. Lastly, we would like to acknowledge our faculty member, Sir Richard, for his support throughout this competition. That is all for our oral presentation. Thank you.